Hi everyone, my name is Madam E.Tavasuria Elangovan. Today I'm going to explain about second part of chapter 6, plasma membrane and transport of molecules. There are three lesson outcomes for chapter 6, but at the end of this lecture, you should be able to understand about the transport proteins and also the transport of small molecules. Before I explain about the transport protein and also about the transport of small molecules, let me do small recap about the previous lesson. In previous lesson, you learned about the fluid mosaic model of the plasma membrane and also the permeability of the plasma membrane. The plasma membrane is actually made up by phospholipid bilayer, in which it consists of hydrophilic head and also hydrophobic tail. Therefore, plasma membrane is very selective. It means only certain ions and also certain molecules can easily pass through the plasma membrane. However, the polar molecules and also the charged ions difficult to across the plasma membrane. Therefore, the transport proteins assist the movement of the polar molecules and also the charged ions across the plasma membrane. Transport proteins actually common with enzymes because they also have the specific binding site for the solute and also can become saturated. Other than that, transport proteins also can be inhibited by the molecules that similar as the normal substrate. So the molecule bind to the transport proteins and they compete with the normal substrate for the transport. Transport protein actually catalyze the physical process, transportation of the molecules across the membrane. There are two types of transport proteins, such as channel protein and also the carrier protein. Okay, so this is the one, the channel protein, and this tree diagram represents the carrier proteins. Channel protein, also known as pore protein. It has a hydrophilic channel which act as a tunnel and also provide the corridors to allow the specific molecule or ions to cross the membrane. For example, the water channel proteins, aquaporins. Aquaporins are channel proteins that allow the water molecules to cross the membrane very quickly. It also play important roles in plant cells, red blood cells, and also certain parts of the kidney. Next is carrier protein or gated channels. Carrier proteins open or close based on the presence or absence of the physical or chemical stimulus. Physical or chemical stimulus is different from the transported molecule. Carrier protein change their shape to move the target molecule from one side of the membrane to the other side. Carrier protein is different from the channel protein because the carrier protein change their shape compared than the channel protein. Both the carrier protein and also the channel protein are specific to the substance that it will translocate. For example, the glucose transport protein in the plasma membrane, which carry the glucose in the blood and needed by the red blood cells for the cellular activities. Glucose transport protein carry the glucose but not the fructose, even though fructose is structural isomer of the glucose because it is very selective. Next subtopic is transport of small molecule. There are two types of transport of small molecules, such as passive and active transport. Passive transport is the movement of the particles across a cell membrane down its concentration gradient without using energy. There are two types of passive transport, such as osmosis and diffusion. Osmosis means diffusion of water molecules across the semi-permeable membrane. However, diffusion means movement of small molecules from an area of high concentration to low concentration. Diffusion can be classified into two types, such as simple diffusion and also the facilitated diffusion. When we learn about osmosis, you should know two important terms such as osmotic pressure and also water potential. Osmosis means the movement of water molecules from high concentration region to low concentration region down their concentration gradient through selectively permeable membrane. If there is a high water concentration, there is a high water potential, but the osmotic pressure is low. Osmotic pressure means a measure of the pressure that must be applied to stop the osmotic movement of water across the membrane. So the water will move from the solution with low osmotic pressure to the solution with high osmotic pressure. Water potential means pressure exerted by freely moving water molecules in a system measured in kilopascal. If there is a high water concentration, there is a high water potential. So the greater the total kinetic energy of water molecules in that system. Movement of the water molecules through osmosis is actually due to the differences in the concentration of solutes in two solutions. There are three types of the osmotic state or tonicity of the solution. Tonicity of solution means the ability of a solution to cause a cell gain or loss of the water. There are three types of the tonicity such as hypertonic solution, hypotonic solution, and isotonic solution. In hypertonic solution, the solution have high concentration of the solutes than the solvent. Hyper means more solutes. Hypo means less solutes, in which the solution have high concentration of the solvent compared than the solutes. Iso means same, in which the concentration of the solutes same as the concentration of the solvent. Imagine that two sugar solutions differing in concentration are separated by a membrane that will allow the water molecules pass through the selectively permeable membrane but not the sugar. Water acts as a solvent, sugar is solute. So there is a two type of the solution, hypo and hypertonic solution. In hypotonic solution, there is a high concentration of the water but less concentration of the solute. In hypertonic solution, there is a high concentration of the solute. It means high concentration of the sugar than the water. So in osmosis process, water molecules move from high concentration region to the low concentration region. So based on this example, the water will move from the left hand side of the glass tube into the right hand side of the glass tube. Therefore, in the second diagram, we can see there is a high level of the water in the right hand side of the U-shaped glass tube. Osmosis and also the water balance between the cells and also the environment is very important to all the living organisms. Let's say if the cell immersed in an isotonic environment, what will happen? There is a no net movement of water across the plasma membrane. Okay, if there is a movement of the water across the plasma membrane, it must be at the same rate in both directions. If cell immersed in a hypertonic solution, in hypertonic solution there is a high solute concentration and a less water concentration. Therefore, water diffuse out of the cell into the surrounding solution and cause the cell shrivel and most probably will die. This condition known as crenation. If let's say the cell immersed in a hypotonic solution, hypotonic solution in which there is a high water concentration than the solute concentration. So the cell gain water, swell and burst. This process known as lysis. There is no problem for the cell or any organism that live in an isotonic environment. For example, the marine invertebrates and also the land animals which embedded in an extracellular fluid that is isotonic to the cells. But the organisms that without the rigid walls, they have the osmotic problems if they stay in the hyper or hypotonic environment because they cannot tolerate the excessive uptake or loss of the water. So they must have the adaptation for the osmoregulation to maintain their internal environment. For example, paramecium, a protease, is hypertonic when compared to the pond water in which it lives. It means the pond water is hypotonic. Even though the cell membrane is less permeable to water than other cells, the water still continuously enters the paramecium cell. To solve this problem, paramecium have a specialized organelle that is contractile vacuole. Contractile vacuole pump to force water out of the cell. The cells of plants, prokaryotes, fungi and some protists have walls that contribute to the cell's water balance. Okay, what will happen if the plant cell or the animal cell placed inside the hypotonic solution, hypertonic solution and isotonic solution? This is the overview of the animal cell and plant cell when they place inside the hypotonic, isotonic and hypertonic solution. If the plant cell placed inside the hypotonic solution, water diffuse into the plant cell and cause the plant cell expand. Plant cell is turgid. This is still the healthy state for the most of the plants. If the plant cell placed inside the isotonic solution, there is no net movement of the water, so the plant cell is flaccid. The plant cell may be wilt. If the plant cell placed inside the hypertonic solution, water diffuse out of the plant cell and cause the cell membrane pull away from the cell wall. So the plant cell is undergo plasmolysis. What will happen to the animal cell? If animal cell placed inside the hypotonic solution, water diffuse into the animal cell and cause the animal cell expand and burst. So this condition known as lysis. If the animal cell placed inside the isotonic solution, there is no changes in the shape of the animal cell. If the animal cell placed inside the hypertonic solution, water diffuse out of the animal cell and cause the cell shriveled. Okay, so we look at the red blood cell. Red blood cells are the examples of the animal cells. So if the red blood cells placed inside the hypotonic solution, we can see the movement of the water into the red blood cell and the cause red blood cell to expand. If the red blood cell placed inside the isotonic solution, there is no changes on the shape of the red blood cells. If the red blood cells placed inside the hypertonic solution, we can see the movement of the water. Water diffuse out of the red blood cell and cause the red blood cell shrivel. Next is diffusion. Diffusion process is one of the passive transport in which substance will diffuse from the high concentrated region to the low concentration region down their concentration gradient. And the molecule or the substance, they use their own kinetic energy. They does not require energy from the cell. So this is the examples of the diffusion of one solute. And this is the example of diffusion of two solutes. Each substance diffuses down their own concentration gradient and not affected by the concentration gradient of other substances. Diffusion can be classified into two types, such as simple diffusion and facilitated diffusion. Simple diffusion means diffusion of small substance without assistance of transport proteins. However, facilitated diffusion means diffusion of small substance with assistance of transport proteins. It means the charged molecules and the polar molecules, they need the transport protein to cross the plasma membrane. Uncharged molecules and non-polar molecules such as steroid, vitamin A, D, E, K, carbon dioxide and oxygen, they can cross the plasma membrane without the transport proteins. Based on this animation, we can see clearly the movement of the substance if they diffuse through the simple diffusion and also if they diffuse through the facilitated diffusion. The molecules that diffuse through the simple diffusion can easily pass through the plasma membrane. However, the substance of the molecules that undergo the facilitated diffusion need the transport protein to cross the plasma membrane. The second type of the transport of small molecule is active transport. Active transport means the movement of particles against their concentration gradient from low to high concentration using the cellular energy obtained from the ATP. So they get the ATP energy from the mitochondria. This type of the transport, they need the transport protein, such as the carrier protein. Active transport is critical for a cell to maintain its internal concentration of small molecules that would otherwise diffuse across the membrane. For example, the liver cells are able to accumulate the glucose molecules from the blood plasma even though the glucose concentration is often higher inside the liver cells than in the plasma. In active transport, the sodium-potassium pump actively maintains the gradient of the sodium and potassium ions across the membrane. So the sodium-potassium pump, they use the ATP energy to pump 3 sodium ions out of the cell membrane and 2 potassium ions into the cell membrane. Generally, the animal cell has high concentration of the potassium ions and low concentration of the sodium ions inside the cell. Let me explain about the active transport. Sodium-potassium pump maintain the gradient of the sodium ions and also the potassium ions across the plasma membrane. So the first step in the active transport is binding of 3 sodium ions into the carrier protein stimulate the phosphorylation of the transport protein by ATP. So the ATP undergo hydrolysis and release ADP and inorganic phosphate. Inorganic phosphate bind and phosphorylate the transport protein. So the transport protein undergo conformational change and then the 3 sodium ions released to the outside of the plasma membrane. As the 3 sodium ions released to the outside of the plasma membrane, 2 potassium ions from the extracellular fluids bind into the carrier protein. This binding cause release of the inorganic phosphate from the transport protein. So the release of the phosphate group from the transport protein cause conformational change of the transport protein and the transport protein back to their original conformation. Then the 2 potassium ions released into the plasma membrane. As the 2 potassium ions release into the plasma membrane, 3 sodium ions bind to the carrier protein and the cycle repeats. This is animation of active transport. We can see clearly the binding of the sodium ions stimulate the phosphorylation of the carrier protein by ATP, then sodium ions released to the outside. 2 potassium ions from the outside bind to the carrier protein and then the carrier protein undergo the dephosphorylation and then 2 potassium ions release into the plasma membrane. So based on the active transport, the molecules move from low concentration region to the high concentration region against their concentration gradient and use ATP energy. So the sodium ions move from inside to the outside, in which inside of the cell there is a low concentration of the sodium ions and there is a high concentration of the potassium ions. At the outside, there is a high concentration of the sodium ions but low concentration of the potassium ions. As a summary, in this lecture, we learned about 2 types of transport of small molecules, such as passive transport and active transport. There are 3 types of passive transport, such as simple diffusion, facilitated diffusion, and osmosis. Molecules that undergo the passive transport move from high concentration region to the low concentration region down their concentration gradient. However, the molecule that undergo the active transport move from low concentration region to the high concentration region against their concentration gradient and use the ATP energy. But the passive transport does not use the ATP energy. The molecules that undergo the facilitated diffusion need carrier or channel protein. Active transport also, the molecule need the carrier protein. That's all from me. Thank you.